glycolysis means the breakdown of glucose. Glyco means glucose, and lysis means breakdown. It's also known as the EMP pathway, and it's the conversion of one molecule of 6-carbon glucose into 2 molecules of 3-carbon pyruvate. And this is the overall reaction for glycolysis. Now, glycolysis is a 10-step pathway that can be divided into 2 phases. The first phase is the energy-requiring phase, or the preparative phase, and the first 5 reactions will fall into this phase. The second phase is known as the energy-generating phase, or the payoff phase, and the last 5 reactions will fall into this phase. Now, before glycolysis, glucose has to enter the cell through blood, a glucose transporter. This is because glucose is water-soluble, so it cannot diffuse directly through the phospholipid bilayer and has to enter through a special transporter. Now, in the first step of glycolysis, glucose is converted to glucose 6-phosphate in the phosphorylation reaction. Now, magnesium ions are important for this reaction as they shield the negative charge on ATP's phosphate groups, which makes the phosphorylation possible. The enzyme hexokinase or glucokinase in the liver catalyzes this reaction, and this is an example of substrate-level phosphorylation. But why do we even phosphorylate glucose in the first place? Well, you must note that the GLUT transporter is bidirectional, so glucose can both enter and leave the cell, but once glucose is phosphorylated, it cannot leave the cell. Now, step 2 is an isomerization reaction, where we convert an aldose sugar into a ketose sugar. So glucose 6-phosphate is an aldose sugar, and fructose 6-phosphate is a ketose sugar. The carbonyl on the glucose on the first carbon migrates to the second carbon, and this is catalyzed by the enzyme phosphoglucoisomerase. Essentially, we are converting an aldose into a ketose. That's all this reaction is. Now, reaction 3 is the phosphorylation of fructose 6-phosphate by the enzyme PFK1, which is phosphofructokinase. Now, magnesium ions are also necessary for this reaction, and this is very important because this is the rate-limiting step of glycolysis, so make sure you know that. Now, the fourth step is the cleavage of a 6-carbon fructose 1,6-bisphosphate into 2 3-carbon molecules known as DHAP, dihydroxyacetone phosphate, and G3LP, glyceraldehyde 3-phosphate. Now, this reaction is actually reversible and is catalyzed by an enzyme known as aldolase. Now, one important thing to note is that only G3LP is actually used in glycolysis, so we need to convert this DHAP, this dihydroxyacetone phosphate, into G3LP. And this is done using an enzyme called phosphotriose isomerase or triose phosphate isomerase. So the result of the preparatory phase is the production of 2 molecules of glyceraldehyde 3-phosphate from glucose, after we are done converting the DHAP into G3LP. So that's the end of the first phase of glycolysis, the energy-requiring phase or the preparative phase. Now we're going to move on to the energy-generating phase or the payoff phase. So G3LP, in fact 2 of them per glucose, is converted to a molecule known as 1,3-bisphosphoglycerate. Now, this is a reversible reaction. It's catalyzed by G3LP dehydrogenase, and even though this is actually a phosphorylation reaction, we are gaining a phosphate, there is no kinase involved. NAD is required for this reaction, and an inorganic phosphate that's present in solution will also be used, but there is no kinase present here. The next step involves the 1,3-bisphosphoglycerate being converted to just 3-phosphoglycerate. Now, this is because there are 2 phosphate groups which are both negatively charged in quite close proximity in 1,3-bisphosphoglycerate, which makes the 1,3-bisphosphoglycerate quite an unstable molecule, so it wants to become something else, it doesn't want to be 1,3-bisphosphoglycerate. So it's converted to 3-phosphoglycerate using the enzyme phosphoglycerate kinase. Oh, but hold on. We thought kinase was for phosphorylation. Why is a kinase being used to remove a phosphate? Well, sometimes enzymes are named for the reverse reaction too, and if you think about it, the reverse reaction is indeed a phosphorylation. So hence the name phosphoglycerate kinase it makes sense. Step 8 is just the repositioning of a phosphate group from the 3rd carbon in 3-phosphoglycerate to the 2nd carbon in 2-phosphoglycerate, and this is catalyzed by a mutase enzyme, as we're changing the position of a phosphate group. In this case, it is phosphoglycerate mutase, as we're changing the position of the phosphate group from 3 to 2. This is also a reversible reaction. Now, step 9 is catalyzed by enolase, and it's also inhibited by fluoride. So this is the reaction that takes 2-phosphoglycerate and converts it into 2-phosphoenolpyruvate. And finally, the last step of glycolysis is the conversion of 2-phosphoenolpyruvate to just pyruvate. This is an example of substrate level phosphorylation, and it is the final step of glycolysis, also the final step of the energy generating phase. So finally, we are done with glycolysis. We have gained 2 ATPs because we've produced 4 and utilized 2, and also gained 2 NADHs, and this is all per glucose. So for 1 molecule of glucose, we've gained 2 ATPs and 2 NADHs. Now, a lot of multiple choice questions like to ask, in the presence of oxygen, what is the total number of ATP produced in glycolysis? And the answer to this is actually 8, because we have gained 2 ATP, but each of those NAD is actually equivalent to 3 ATP, so the answer would be 8 if you are ever asked this in a multiple choice. So moving on to the questions that have been asked. The end product of glycolysis is one of the following. The answer is pyruvic acid. Now, glycolysis produces 2 molecules of pyruvate. The terms pyruvate and pyruvic acid are used almost interchangeably in biochemistry. Pyruvate is just the conjugate base to pyruvic acid, which is what predominates at a physiological pH. So pyruvate predominates at a physiological pH. EMP can produce a total of how many ATP? Well, EMP just refers to glycolysis because EMP stands for the Embden-Meyerhof-Parnas pathway, and it produces a total of 8. Now, this is a bit of an interesting question because it's talking about anaerobic respiration, not aerobic. So the net production of ATP by glycolysis in anaerobic respiration is one of the following. The answer is 2, because we can't use the NADH to produce ATP, as the NADH is used to reduce the pyruvate into lactate, as you can see in this diagram. The next question is just hexokinase is dependent on which of these? The answer is magnesium, and this is because magnesium ions shield the negative charge on ATP's phosphate groups, which is what makes the phosphorylation possible. If you remember that, hexokinase is the enzyme that catalyzes the conversion of glucose to glucose 6-phosphate. During which of the following steps of glycolysis does phosphorylation not occur? The answer is B, the conversion of glucose 6-phosphate to fructose 6-phosphate. This is because this is simply an isomerization reaction, converting an aldose sugar into a ketose sugar. Nothing else. There is no phosphorylation involved here. It's just an isomerization. Which of these is an energy utilizing step in glycolysis? It is D, the conversion of fructose 6-phosphate to fructose 1,6-bisphosphate. Now, while the first phase of glycolysis, in which the first 5 reactions are passive, while this is the energy requiring phase or the preparative phase, only 2 reactions out of the 5 actually consume ATP, and this is one of them. So if you have any questions, drop them in the comments. Hopefully this video has explained something about glycolysis. Thank you for watching.